Welcome to the Forest School Podcast, a show where I, Lewis, and my co-host, Wem Southerner, set out to explore a shared pedagogy. It's a space where cerebral thinking matters more than craptivities, where educational purpose outweighs pretty resources, and where inclusion is more than just a buzzword. Join us as we overthink the trivial, deep dive into whatever sparks our dopamine, and reflect on what it means to truly hold space for others, holistically and without judgment. Thanks for being here. We hope you enjoyed this episode. Hello, welcome to the Forest School Podcast. It is me, Lewis. I'm recording the intro on my own today. Wem is off doing some other things. Thank you so much for joining us. In this week's episode, we are talking to Dr. Richard Buggs, who works at the Royal Kew Gardens and also at Queen Mary University in London, about some of his incredible work in various different ways, talking about him sequencing the DNA of various different trees. Specifically, we wanted to talk to Dr. Richard Buggs about the fact that in Kew Gardens, they have started finding more and more ash trees that are resistant to ash dieback, and kind of like what that means for the future of ash trees, particularly in the UK, what it means for biosecurity going forwards, all these kind of juicy, incredible, sciencey things. He explains everything so well. We had a couple of technical issues, and we're so glad that he managed to come back in halfway through the episode, because it was like, he was explaining things in such a beautiful way, and it makes so much more sense to hear someone who really knows their stuff in a scientific way explain it to two like cruddy pleb people that are me and Wem, covered in mud, going like, so trees and you do that, and he just explains things in this amazing way, and then right at the end of the episode he was very kind and gave us a little bit of extra time to talk about Darwin's monstrosity, which is all about this incredible part of Darwin's Origin of Species that doesn't quite make sense, and it didn't quite make sense when he wrote it, and now even more evidence has come up which makes it sort of make even less sense now than it did back then. And it's so interesting to think like, there's still stuff we don't understand. So that's right at the end of the episode. Thank you to everybody that's supporting us on Patreon. Really appreciate it. It means the world, not just financially but also emotionally. It's so great to hear from you people, and because we don't hear back, we're sort of talking into, like I'm doing right now, talking into the ether. So it's lovely to hear the stuff coming back on social media, on Patreon, anywhere that you guys are getting in touch with us. We really enjoy and appreciate the messages. So without further ado, let's get into our chat with Dr. Richard Buggs.
So we're here today with Richard Buggs. Thank you so much for joining us, Richard. We first came across your work through an Instagram post that Kew Gardens put out about the research that's happening in conjunction with them and the university where you're based about ash dieback. And we thought, that's such a juicy topic. We'd really love to speak with you. So we reached out and you very kindly agreed to give us some of your time. But could we start off by you just telling us a little bit about your career and your work and what you do?
Well, thanks for having me. It's great to be here. So I work at two places, Queen Mary University of London and Royal Botanic Gardens, Kew. And I do research for both of them on the same topic, and that is the genomics of broadleaved trees. So that's studying the DNA of trees, mainly in the UK, and we're specifically interested in threats to them from pests and pathogens, and also from climate change. So what we're trying to understand is how the genetic diversity of our tree populations can help them to resist new threats, and how we can do things to help them help themselves.
That's
so juicy.
So juicy. And I saw on your website that you've actually got a species of birch named after you.
That's right.
Buggsii.
Yeah.
Am I saying that right?
Betula buggsii.
Can you tell
Yes.
us how that came about?
Well, I've been working on birch for quite a long time, longer than any other tree that I've worked on. And I had a PhD student from China. He was actually my first ever PhD student, and he worked with me on birch. And then he went back to China and started his own lab at the Shandong Agricultural University. And he discovered a new species of birch in China. And to my surprise, he decided to name it after me. And so it's Betula. Betula is the genus birch. Buggsii, that means of bugs. Yeah, it's quite, all through my life, my friends have called me Buggsii, so it's quite funny to have a tree called Buggsii.
Do you have one? Like, have you got a Betula buggsii
No, no, I'm trying to get one, but it's quite hard to bring the seed into the UK for good reasons, because of biosecurity and sharing of plant genetic resources. It's actually quite difficult these days to bring a new species into the UK. But once we can get all the paperwork done, I'm hoping to get some seed and maybe have one in my garden.
Yeah.
That'd be so cool.
So does it currently only grow in China?
Currently, as far as we know, it only grows in China. Yeah.
Richard, can I ask? So your work is about like genomes and sequencing genomes, and I saw in some of your research that you've done like 2,000 oak trees. How do you arrive at that as a kind of project? Had you come from like a horticultural arboristy type background and then moved into the scientific side of things, or did you start in the science and kind of go, oh, this particular area is very interesting? How do you end up where you are now?
Yeah, very much started on the science side of things. So going right back to school, when I chose my A levels, I wanted to be a vet. So I mainly chose the sciences. Then partway through my A levels, I decided actually I don't want to be a vet. Ironically, partly because you have to spend so long at university to become a vet, and I wasn't sure if I wanted to spend that long at university. I've then spent the rest of my life in a university. But anyway, there you go. So I decided not to be a vet and ended up doing natural sciences at Cambridge and specialised in plant sciences at the end, which I really enjoyed. Then went on from there to do a PhD at Oxford in plant sciences again, working on what is essentially a weed called annual mercury, trying to understand its population dynamics. Then I did postdoctoral research at the University of Florida on the evolutionary genetics of trees, looking at a new species that arose just about 100 years ago in America through the hybridization of two species of a plant called Tragopogon, which arrived from Europe with European settlers. And there they hybridized and made a new species. So I studied that for a bit. Then I moved to Queen Mary University of London as a research fellow and started to work on birch. And at the time, I just chose birch because it was a useful system for studying the kind of things I wanted to study. So it was very much theoretically driven. But while I was studying birch, the ash dieback problem started. Ash dieback was discovered in the UK in 2012. It's a fungus that's not native to here. It's an invasive fungus. And suddenly everyone was interested in ash trees. And I was one of the few people in the country at the time working on the genomics of broadleaf trees. So I applied for funding from the Natural Sciences, from the Natural Environment Research Council to sequence the genome of ash. And then that launched a larger research program on ash genomics that I'm still actually working on today, 12 years later. But after some of the early successes of that program, I began to be asked to work on other species as well. So in the example of oak that you mentioned, a researcher called Sandra Denman at Forest Research had spent many years studying a problem that oak trees have called acute oak decline. And she'd done lots of work on different aspects of this disease that is killing oak trees. And she said to me, could you come and do some work on the genetics of this, see if there's a genetic aspect to it? So I said, yes, sure, but it'd be very expensive because we'd have to sequence the genomes of loads of oak trees. But thankfully, DEFRA, the Department for Environment, Food and Rural Affairs, stepped in with some funding. And so over the years, we've managed to build up this data set of about 2,000 oak trees that we've got the whole genome sequence of. And that's enabled us to work out how much of a genetic component there is to acute oak decline. And it turns out it's quite a small genetic component. There is some genetic aspect to it, but it's mainly determined by the environment that the oak trees are in. So that's a really valuable piece of information that helps us to manage acute oak decline, because it means we can do things to try and help the environment of the trees to help them overcome it. We don't so much need to be trying to breed oak trees to have resistance to acute oak decline.
Can I ask a question that might be too difficult to answer in layman's terms, but how do you sequence the genome of a tree?
Well, basically,
we're trying to work out how do you ask that?
Yeah, we use techniques that were first developed for the Human Genome Project. Lots of the methods we used were actually first developed for humans, and we now apply them to trees. First first of all, we have to collect a sample, then we extract the DNA from the sample, and you can do some clever chemistry in the lab to extract the DNA. That's quite routine. And then we send it off to sequencing centres, which are these massive places like factories that can process thousands of samples, again, mainly set up for human research. But of course, once you've extracted DNA is the same in terms of its chemical composition, whether you're a tree or a human. It's just that the sequence of the letters is different. The information that it contains, of course, is very different. And at these sequencing centres, they have massive machines that use very clever technology that essentially, one by one, causes the different letters of the DNA to glow in a different colour. And it just works through sort of essentially photographing flashes of light as the different letters glow. And by interpreting the sequence of the flashes of colour, it can calculate the sequence of the DNA letters, essentially. But it only does that for fairly short fragments. So we get back huge data sets that are all made up of tiny fragments of DNA, which are essentially pieces of a jigsaw puzzle. And then we have to use a supercomputer to make sense of all that and bring it all together into a meaningful data set that is telling us what is the genome of a particular individual.
Wow. And when you say you collect a sample, does that have to be wood, or can that be a leaf or sap, or does it matter?
Normally we use leaves, but sometimes if we can't get leaves, sometimes we'd use the cambial tissue, which is the bit of tissue just under the bark. That's quite often got a lot of DNA in it. Sometimes we use buds of the tree if we're having to sample in the winter. And one of my colleagues at Kew has just started to extract DNA from pollen. You can also collect pollen and extract DNA from that.
And you've mentioned a few times that you were researching broadleaf trees in general before this. Is that because broadleaf trees, they have some sort of similarities in research methods, or that they have linking things? What makes them, I mean, obviously broadleaf trees are their own group, but what makes them kind of a closed group for sort of research purposes?
Well, in a sense, they're not, and actually, they're not really a closed group in a scientific sense at all. In that ash trees are more similar to some shrub species than they are to an oak tree. But I suppose what brings them together is the range of stakeholders is very similar. So the people who are interested in the results of a study on ash are also interested in the results of a study on oak and on elm and on birch, and the people who fund that kind of research are interested in it. So I suppose it's more from a human point of view that these sort of come together. I'm having to interact with the same set of stakeholders and funders and interested parties. Yeah.
When you said stakeholders, the first image that came into my head were animals and birds. Creatures.
Yeah.
I was like, yeah, no, that makes sense that, the same stakeholders are involved, like the same creatures that like live in and around broadleaf trees. And I was like, no, clearly he doesn't mean that. But yeah.
There's truth to that too. I mean, they're in very similar ecosystems a lot of the time. But of course, some of them do support the same insects. Some have insects that are unique to them. And so if you lose that species of tree, you also run the risk of losing that species of insect as well. So, for example, with elm, there's a butterfly called the white letter hair-streak butterfly, which needs elm trees in the landscape. And if we don't have elms, we don't have the white letter hair-streak butterfly. So a lot of conservation of elms at the moment is around the conservation of this butterfly. So there are, you're not actually far wrong in pointing out these other interactions with other forms of life.
Oh, this podcast is made possible thanks to the generous people who support us on Patreon. If you're enjoying the chats that we're doing, if they're improving your practice, or you're just finding them interesting, head to patreon.com/the Forest School Podcast. You can support us from as little as £2 a month, and it really does make a huge amount of difference to keeping the lights on, keeping our bills covered, and us being able to find the time to keep interviewing these cool people, read these incredible books, and keep providing this resource to the community. Thanks so much. Bye.
Are there any species that are specific to ash trees?
Yes, there are about 50 species of not just of insects, but also of fungi and other microorganisms that are reliant on ash trees. Yeah, a big study was done by someone called Ruth Mitchell at the James Hutton Institute in Scotland. About 900 species depend on ash and about 50 depend on it completely. They really, really need it, and it can't be replaced for them.
So this research that you've been doing on ash dieback really is, really, really vital. Can you explain a little bit about ash dieback? You said it was a non-native fungi. How does it affect ash trees, and how does it cause damage to them?
Yeah, so it's a fungus that originally came from East Asia, and in East Asia, it doesn't seem to damage the living trees. It helps break down the leaves once they've fallen off of the trees, but it doesn't seem to attack the living trees themselves. But over here, because our native species of ash hasn't had a long history of coevolution with this fungus, it's susceptible to it and it can attack the living tissue. And it was first noticed in this country in 2012. It had probably arrived before that. And it reproduces in the fallen leaves. So it produces little mushroom-like bodies in the leaf litter on the floor of the forest. Tiny little white mushrooms grow on them. If there's a damp spell in the summer, you can go out into the forest and just scrape away some of the leaves. And if you're in an ash woodland, you'll find these little things that look a bit like twigs, but they're actually the middle of the leaf, because ash have a compound leaf with a stalk down the middle. You can find these stalks from last year. The leaflets have largely fallen off. You just have this stalk, and out of it, it'll be black, but out of it, there'll be these little mushrooms, little white mushrooms, and they produce spores of ash dieback. And when those spores are produced, they then float up in the air and they land on the leaves of living ash trees, where they start to grow, and they grow into the leaf. And if they can, they grow down through the leaf and into the woody tissue and start to attack that. The trees want to stop them, so some trees seem to be able to do things to stop them, but other trees are very susceptible. And the fungus grows down in it, then blocks up the pipes in the tree, and the bit of the tree above the blockage just is starved of water and nutrients and dies. And that's why the disease is called ash dieback, because the ash trees are literally dying back. You can see that the top of the tree just has this layer of dead twigs sticking out the top above the leaf canopy, where the fungus has killed off the woody tissue.
Yeah. And we hear about it a lot in terms of public safety as well, don't we? In terms of ash that's growing near footpaths or rights of way or whatever, being felled, because then they're at risk of falling.
That's right. Yeah, it does become very dangerous, because trees with ash dieback can get very brittle and fall over in unexpected ways, unexpected directions. And sometimes a tree that looks quite healthy will actually have quite a rotten trunk. So you do have to be very careful. So in any area with public access, it is a good idea to be very vigilant about ash dieback. And if a tree is clearly unhealthy, it is wise to fell it, particularly if it's above a forest school, I would say, because you just don't know when it will drop a limb or fall over itself. And unfortunately, the more they have the disease, the more dangerous they are to fell, because even some very experienced chainsaw operators have been caught out. They think they know which direction the tree is going to fall, but because part of the trunk is brittle and part of it isn't, the tree ends up falling in a direction they didn't expect. So you do have to be extremely careful. On the other hand, but if you've got a woodland that doesn't have public access, it's quite a good idea just to let nature take its course. We had a study recently published in Science where we showed that in a woodland that has just been left to naturally regenerate, the younger offspring of the ash trees in the wood have on average higher resistance to ash dieback than the adult trees. And so the population itself is naturally evolving to have greater resistance. And obviously that's great for the long-term future of ash. So where you can, it's good just to leave an ash population undisturbed, maybe protect it from deer and other herbivores, But if it can be left undisturbed, then it's got a higher chance of being able to regenerate and have this next generation, which hopefully will be more resistant.
And so is that then part of you said, some of your funding and stuff comes through DEFRA and people like that. And so is this then a situation where your research kind of provides advice to people like the Forestry Commission and farmers, or is it that specific species that you are, sequencing and growing get kind of shipped out as like, hey, go plant these? What's the next step with this research to kind of get it into woodlands?
Yeah, that's right. So the idea of the research is, first of all, to inform policy and management. So if a new tree disease comes in, the question is, how do we deal with this? Do we try to eliminate it, or is it too late to eliminate it? Is there any natural resistance? Will nature by itself lead to that resistance increasing? If we just do nothing, will nature recover on its own, or do we need to step in and help nature? Either by managing the woodland in a particular way or by breeding trees that have resistance to this new threat. And so with the example of ash dieback, quite early on we realised there is a degree of genetic resistance out there in the ash population. So the future looks brighter than it did for something like elm with Dutch elm disease. And so now the question is, well, to really make use of that genetic variation, do we need to have a breeding programme? Or will natural selection do it for us? And so it's very encouraging to have a result showing that natural selection is working. And the way it's working is simply that there's lots of diversity in the adult population. And every time seed are produced, they have a random mixture of the genes that are found in the adults. And some of those random mixtures combine lots of different pre-existing genetic variants that give greater resistance to ash dieback. And if they combine more of those variants than are combined in any of the parents, then that offspring will actually have more resistance than its parents. So even though it's basically the same gene pool, it's just being mixed up in different ways. And in different individuals, there are more genes for resistance. And the fungus is coming in, and at a very young age, it's killing off the young trees that are not resistant. It can kill the young trees much faster than it can kill the big old trees. And so the younger trees that survive are actually the ones that are really more resistant. So they're a non-random subset of the offspring of the adult trees. And so that's how nature is giving us, just in a single generation, from all of the pre-existing genetic information, it's giving us some offspring, some younger trees that are more resistant to ash dieback.
Are you thinking of working outdoors in a way that's actually authentic and meaningful? Our Forest School Leader course isn't just a conveyor belt of trainees getting trained and sent off into the world with no support. It's something that changes how you see people, community, and nature. Across 10 practical in-person days, you'll pick up everything you need to hold space for people and to facilitate authentically deep and meaningful Forest School experiences. We cover so much stuff on this course, like child development, practical skills, and how to offer a calm, non-judgmental approach that people in nature really need. It's a course that can meet you wherever you are in your learning journey, whether you're already working outside, working in education, or you're somebody who just wants to change. We tailor all of our teaching and learning experiences so that they meet the diverse range of needs of students that are coming to us on any given course. We hope that people finish the course not just qualified, but also feeling secure that they have had the time and space to develop their own personal pedagogy, their own approach to forest school that they can then take with them out into the world. If that sounds like something you'd like to be a part of, then why not come and grab your spot on one of the most inclusive, authentic and meaningful forest school courses around. Search for Children of the Forest Forest School Training online or check out the links in this video's descriptions.
And is it right that this research is a little bit surprising because that evolution is happening quicker than we might expect? Is that right?
Well, I suppose in some ways, if you think of evolution as something that happens really slowly, then it sounds surprising. But actually, this is something that's been predicted for quite a long time that where you have a trait that is determined by lots and lots of different places in the genome, then you would predict actually you could get a very rapid evolution of the trait, because among the random mixtures that come about among the offspring, there are likely to be some that combine a really good combination of the pre-existing information. Yeah. So in some ways it wasn't surprising. I think the reason why it became quite a high-profile publication was because it's been very hard to actually show this in the wild. It's been very hard to show this process of natural selection happening in a completely natural setting. And so that's what was unusual about our finding, I guess, that we were able to demonstrate it at the DNA level in a natural woodland.
Rather than it just being theoretical, it's literally this generation, we are seeing this. Yeah, amazing.
Yeah, so the theory was quite strong and quite persuasive. I mean, and it's kind of, it's really common sense that this should happen. You know, if you imagine a population of humans, and you know, we have traits like height. So human height is determined by thousands of different places in our genome. You know, it's not the case that there's one gene, and if you have one variation of that gene, you're tall, and if you have the other, you're short. And so there are tall people or short people and nothing in between. You know, there's continuous variation in height because it's determined by thousands of different loci. So if you imagine there was a big family, all of the offspring of the family, slightly different heights, then imagine, say, something terrible happened, like there was a war and anyone over 6 foot got recruited into the army and were killed. Well, the surviving offspring from that family would be all the short ones. They would have a non-random set of the genetic variation of their parents, because there's been this removal of part of the family. And that's what's happening with these ash. The offspring of any two individuals of ash are varying in their level of resistance to ash dieback, but then the susceptible ones are just being taken out by the fungus. And so the survivors are the ones that have the most beneficial combination of the genes under this particular selective scenario.
Yeah.
And is that variant something that is going to be in the ash trees? But, you know, is that something that's able for people to see without sequencing the genome from the outset? Are there any other markers that it triggers that means that you could sort of go, oh, that one's going to be quite resistant, or that one's not going to be very resistant? Can you tell it before the fungus gets involved, or is it kind of got to sequence it?
Yeah, the reason why no one's been able to show this before is that you do have to be working at the DNA level. So with more old-fashioned methods that could have been done before we had these new technologies, what you would have had to have done was you would have had to have taken each of those adult trees and each of those juvenile trees. You'd have had to have cloned them. You'd have to have planted them out in trials and then monitored them over several years to actually work out if the younger generation really do have higher genetic resistance than the old generation. It'd be a really, really hard, laborious thing to do. But because these days we can actually sequence the genomes and look at what's happening at the DNA level, we were able to show that this is happening.
Amazing. So how does that, are there differences or similarities there with Dutch elm disease and how elms may or may not be resistant to that? What's the future looking like there?
Yeah, there's definite similarities in that this is a disease that's coming from outside. It's started to kill off loads of trees. I think for elm, things are a lot worse for a couple of reasons. First of all, a lot of elms in our landscape are clones planted by humans. So a lot of the really classic English elms that used to be over the British landscape, that were a really sort of important part of the landscape, a lot of them were all exactly the same genotype and have been planted by humans. And so there's not the genetic diversity there in the same way as there is for ash. And in addition, elm don't reproduce very much by seeds, and that particular clone didn't reproduce at all through seeds. So the elm aren't naturally in Britain doing, or they're very rarely doing, what the ash are doing very commonly, which is mixing up all their genes through sexual reproduction. Elm also have the disadvantage that they're not killed by the disease until they're about 12 years old. And so it takes a long time before natural selection can start to act, whereas with ash, they seem to be susceptible very young. And so you can get a whole carpet of seedlings, and then the most susceptible get taken out. And then the ones that survive can use all the nutrients and the space and the light. And so there's a real advantage there for ash in the way that the disease is working.
This is like blowing my mind. This is so interesting. How do you, one question at a time, right? How did people clone the elm trees that were everywhere, and how were they reproducing if they weren't reproducing by seed?
Cuttings,
Yeah.
right? Surely.
Yeah, cuttings or suckers. Yeah.
Yeah. Okay. So if
And so
humans
that's
hadn't,
exactly the same tree every time. It's just
Yeah.
so if humans
Yeah.
hadn't mucked about with it, say humans hadn't taken cuttings or anything like that, would elm, like way back when, would it have reproduced by sucker and by seed if we just like not interfered? Is that right?
Yeah, to some extent. So we actually have two elm species mainly in the UK, wych elm and field elm. Wych elm does reproduce naturally through seeds. Field elm doesn't very much. And the particular fast-growing massive tree was just a single clone. I guess it was chosen because it grows fast. And I think part of its fast growth and big growth was actually because it's not reproducing at all by seed or pollen. So all of its energy can go into growth.
Perhaps I could ask one question to kind of maybe finish off, which is about climate change and other threats to trees, as you said you've been researching. Is there kind of like an overall finding that you're coming across in terms of like human interference? Are we learning lessons from this research and from looking at Dutch elm and ash dieback in terms of how we should be behaving around trees to avoid what's happened with elm and to hopefully give trees a better chance of survival?
Yeah, one thing that strikes me is that the threat to trees from new pests and pathogens is happening far quicker than the threat of climate change. You know, when you look at the devastation that ash dieback has caused to our landscape over just 12 years, it's hugely greater than the effect of climate change over those 12 years. And so I think we just need to be so vigilant about pests and pathogens and put energy and resources into preventing new pests and pathogens from coming into the country. So DEFRA have a risk register of plant diseases and pests that are not yet in the country but could come, and it's quite a long list. And we need to just be so careful that we do keep these other things out of the country, and that when we do trade agreements, we're not compromising biosecurity for our plants. Because we see every day the devastation that these accidental introductions can cause.
I was going to say, well, accidental is an interesting word. I was going to ask Richard what your view is broadly on like Victorian botanists and how you feel about they've had an impact on the UK. Because you said accidental, and some of these things are not accidental, right?
Well, I suppose when it comes to invasive plant species like rhododendron, they weren't accidentally introduced, but it was accidental that they began to invade the understory of our woodlands and were so hard to get rid of. I think with pests and pathogens, it is normally accidental because no one is wanting them in their garden or anything like that. So, we just need to be so careful about these things. And today we move live plants around the world far more than people used to. So Victorian botanists were generally moving around seeds, I understand, whereas today we can have a large tree growing in a pot and just move it in a shipping container anywhere in the world in a way that couldn't really be done or wasn't done so much before.
Is there anything, I guess as a last question, that those of us who are responsible for woodlands or running forest school in woodland often, is there anything that we can do at our level to help prevent this kind of thing happening, or from, if we're thinking about sourcing trees or saplings from particular places, or is it all kind of like above the level that we're kind of working at?
Well, I think we can be careful about where our trees are sourced from, and it's better to be buying saplings that are grown in Britain rather than grown abroad. If someone comes around to your house with a white van and offers to sell you an olive tree out the back of their white van, it's best to say no. There are decisions that we can be making in our day-to-day life about plant biosecurity. and trying not to spread infections between woodlands if we possibly can. But then with something like ash dieback, it's already everywhere, so there's very little we can do to prevent its spread. I think the main thing is to protect regenerating ash seedlings from deer. That's probably the biggest threat to ash now.
Yeah. Oh, thank you so much. This is it's been an absolute joy to speak to you. As you can probably tell, I'm very excited, and we could have talked for 20 hours about many other things. But thank you, Richard.
Do you want to talk about Darwin's Abominable Mystery quickly, or?
Yeah, go on.
Yes,
Ben.
if we've got time. Yes, please.
Since you've mentioned it now, let's talk about Darwin's Abominable Mystery. What is it?
Well, so Darwin's theory is all about natural selection, and so we've demonstrated that happening in a natural woodland from pre-existing genetic information. But what we're not showing in our study is anything actually new arising. We've just got new combinations of what was pre-existing. These ash trees are still ash trees. They've still got all of the same gene pool as their parents. They've just got a subset of it that is non-random. But Darwin realised that for natural selection to actually make anything new, it would have to happen really, really, really slowly. And so he expected, from his theory, you'd expect everything to happen really gradually. And so he predicted that in the fossil record, you would see these very gradual changes. But the problem for him was that you don't see that in the fossil record. And he had to spend 2 chapters of The Origin of Species basically trying to explain away the fossil record. And one of the aspects of the fossil record that really came into focus, mainly after he'd published The Origin of Species, just in the last decade of his life, was the fossil record of plants. And it turned out that flowering plants, which we sometimes call the angiosperms, they suddenly appear in the Cretaceous. And they appear with no apparent sort of antecedents. You suddenly get this new form of plant appearing in diversity. And so this was such a puzzle to Darwin that when he was writing a letter to Joseph Hooker, who was the director of Kew Gardens at the time and a great friend of his, he said that this is an abominable mystery because it was so out of keep with his theory. And that phrase abominable mystery is quite often used by scientists working in this area to talk about that aspect of the plant fossil record. And so it's something that I'm very interested in. And I did a bit of historical research on what Darwin meant by it at the time. And it turns out that he was actually referring to a slightly smaller group of plants than we refer to today when we refer to the abominable mystery. So in some ways, the abominable mystery has actually got more mysterious over time, not less mysterious. And that's quite fascinating. More abominable, yeah. And that's quite interesting because science is often presented as something that is just constantly dispelling mysteries and constantly solving things. But actually, some of these mysteries are pretty intransigent. They hang around. And so that's something that I have a side interest in.
Wow. And is it something you're exploring? Do you have a personal... would you be willing to say, do you have a personal kind of this is what I reckon happened, or this is what my research hopes to point towards, or is it just kind of beautiful because it's unexplained?
Yeah, it's beautiful because it's unexplained. I suppose where I differ from Darwin is, so he thinks of something like that as an abominable mystery. I think I'd probably see it more as a sacred mystery. So unlike Darwin, I believe in God and I'm a Christian. And so I'm open to looking at something like that and saying, okay, this is a sacred mystery. I'm open to the idea that God could actually have intervened and done something at that point in history, which of course was what Darwin was totally against. So I'd be more sort of inclined to call it a sacred mystery rather than an abominable.
I think I've also got another idea. What I just think I might have solved this mystery actually, an alien pod.
Oh no, stop.
From another planet, and it had some seeds of plants on it, and it landed, and that's proof that aliens exist because they sent the pod of things.
People have suggested that kind of thing, particularly more for the origin of life than for for the abominable mystery. But you would be surprised how many serious scientists might think that kind of thing for the origin of life. But how you distinguish that from God is, how do you know? How would you tell the two apart?
Or even a bit of another planet that just like, you know, a meteor or something and a bit of earth from another planet with some seeds embedded in it came to our, oh my, this is so amazing. Oh my gosh.
But what's difficult there is you're kind of pushing the problem back. You've still got to explain how they came about on that other planet. You've still got to explain how the alien civilization arose. But the great thing about God is he is the uncaused cause. You know, down through history, whenever people have talked about God, he is the end of that infinite regress, in that he always is. It's a neater solution, I think.
What are the odds that loads of dinosaurs were into floristry and we're going to find lots of bouquets in a different fossil record somewhere where they've made lovely arrangements?
Well, that would certainly make headlines in the scientific literature. That's for sure.
Richard, I'm so glad we got that in. That's lovely. I really enjoy it. It's so nice to know that there are still like amazing breakthroughs happening and kind of still mysteries and kind of wonder and awe and, you know,
absolutely,
all those things to still be found in the world.
And we just have to look after the natural worlds around us and make sure that we don't lose it.
Absolutely. Thank you so much. If people want to find out more about your work and what you're up to, where can people go to find out about your stuff?
I have a website, just www.richardbuggs.com, and that links through to my university site, etc. So feel free. And I'm on social media as well. My name is very easy to find.
Yeah. Buggs with two G's. Two G's.
With two G's.
Yeah.
Brilliant.
Second G is
so much,
very
Richard.
Yeah.
Thanks for having me.
Thank
and thanks
you
for the
so
work
much.
you do with Forest School. It's brilliant.
Cheers.